Hello everyone, this is Dr. Susan from the Radiological Technique Department. The goal of today's training program is about preparation of mouse embryonic fibroblast cells suitable for biomedical research. This is first, and second, clarify the importance and application of MEF cells in biomedical research in general. So first question is, what are MEF cells? MEFs are a type of fibroblast prepared from mouse embryo. MEFs, as we can see from the picture A and B, are always shown a spindle shape when cultured in vitro. And as I mentioned, MEFs are a type of fibroblasts. So as a reminder, what are the fibroblasts? Fibroblasts are a group of heterogeneous resident cells of mesenchymal origin that have various locations, diverse appearances, and distinctive activities. These cells are poised to respond to factors released by newly activated innate immune cells, thus becoming a useful tool to study inflammation and immunity. So before we get to the protocol and how we isolate the MEF cells, we have to mention the importance of these cells. So basically, as far as we know, genetically manipulated transgenic and gene-targeted mouse models are indispensable tools used in defining the role of genes in developing an organ function. Similarly, cells isolated from these mice can have the same genetic alterations and therefore can be extremely useful for studying the molecular and cellular mechanisms and regulatory gene network involving an imitated gene under well-defined culture conditions. So this slide shows a schematic representation of MEFs isolation procedure. So basically we will start from the first step and dissecting sacrificing the pregnant mice, and then we dissect out the uterine horns, and then we briefly rinse the uterine horns in 70% ethanol and then wash it with PBS. And we can do these first two steps in non-aseptic condition, and then we can later separate each embryo from the placenta and the embryonic sac and remove the head and the red organs and wash the remaining parts or tissues from the embryo in PBS. And then we place all the embryos in a clean petri dish. And then we will show how we mince the tissue using a sterile razor blade until it becomes pipetable. And then we add a trypsin and DNase 1 to help digesting the remaining tissues. And then we incubate the tissues for 15 minutes at 37 Celsius. And then we, during this incubation time, we try to pipette up and down after each 5 minutes in incubation. And then as I said in here in this step, we add the trypsin. We know that trypsin help in digesting the tissues, but we don't want all the tissue to be digested and all the cells died. So we have to inactivate the trypsin by add MEF medium. And then we centrifuge at very low speed for 5 minutes, and then we remove the supernated and all the tissue that is not minced with the supernated, and we keep the pellets. And then we resuspend the cell pellet in MEF medium again, and we try to place 3 to 4 embryos in each flask coated with gelatin, and this called passage 0. We can freeze it in liquid nitrogen, and we can keep culturing that until we get to passage 3 or 4. So starting from this slide, we are going to get into the isolation of mouse embryonic fibroblast process in details. So as a reminder, the first two steps are performed under non-aseptic condition. Those steps are the first one, sacrifice a pregnant mouse at 13 or 14 days postcoitum by cervical dislocation. Basically, sacrificing the pregnant mouse can be performed by putting the pregnant mouse inside a container has only CO2, and it takes a few seconds, maybe 30 seconds to 1 minute for the mouse to pass away or die. The second step is dissecting out the uterine horn briefly. So basically, after the mouse pass away, so we start dissecting the mice and take out the uterine horns and rinse it in 70% ethanol. As we can see from this picture, we are putting, this is the uterine horn. We place it in 70% ethanol, and after that we wash it with PBS. And we have to notice that the PBS should have no calcium and magnesium. And this is how we place it. The following steps are carried out in a tissue culture hood under aseptic condition and using sterile instrument to avoid the contamination. So basically, we have now the uterine horn sterilized and washed with PBS. So what we are going to do, we put the uterine horn into a petri dish and separate each embryo from its placenta and embryonic sac. So basically we cut the first embryo, and as we can see, this is the first one, and this is the surrounding embryonic sac. We have to remove it and remove the placenta as well. And this is how it looks like after we're removing the sac. And this is the embryo, this is the head, and this is the tail, and this is the red tissues. So after that, what we need to do is dissect the head and the red organs. So we have to remove the head and the red organs, this is the blood, and wash it with PBS. As we can see, so after we remove the head and the red organs, the remaining tissue of the embryo, we have to wash it in PBS and place all the embryo in a clean petri dish. And so we can finally mince the tissue with sterile razor blade until it's become possible to pipette. So here, after we rinse, we move to the next step, which is mince the tissue using sterile razor blade. And this is the blade, and this is the petri dish, and we have all the tissues from the embryo. So we have to mince it. Okay, and so a closer look for how to mince the tissue, and we keep mincing until it becomes possible to pipette these tissues. After that, what we are going to do after we mince it very well, we add 1 mole of trypsin EDTA, and including the DNase 1 to help digesting the tissues. Okay, and after that's what we are going to do to transfer the tissue into 50 ml Falcon tube and incubate it in water bath for 15 minutes at 37 Celsius. And during the incubation, we have to keep pipetting up and down thoroughly to help dissociation of the cells and prevent clotting. So this is the water bath, and we put our cells for 15 minutes at 37 Celsius, and we don't forget to pipette up and down each 5 minutes. And here the next step, so we inactivate the trypsin by adding about 1 volume of freshly prepared MEF medium. So basically in the previous step we add the trypsin to help digesting the tissues, so we don't want the tissues to be digested or the cell die, so we have to inactivating the trypsin by adding MEF medium. The MEF culture medium, basically it's a mixture of all components. All the components are fertile. Those components are 450 ml of PMM and 50 ml of FBS 10% and 5 ml of L-glutamine and 5 ml of penicillin to avoid the contaminations. So here, after we adding inactivating the trypsin, we do centrifugation at very low speed for 5 minutes, and after the centrifugation finish, we remove the supernated and resuspend the cell pellet in warm MEF medium again. So here this is a step showing how we remove carefully all the supernated and discard it. Here they showing how we resuspend the cells in warm MEF medium. Okay, warm. So So this slide showing we plate approximately a number of cells which is equivalent to 3 to 4 embryos in each flask coated with 0.2% gelatin for 2 hours. The fibroblast is called passage 0 or P0. Okay. Those cells are the only cells that have the ability to attach to the gelatin coated flask. We wait 24 hours, and after 24 hours we will watch the cells and check them, and we will notice that they will reach 80 to 90% confluency, and at this stage we call them MEFs, passage 0, and those cells can be froze for future use, or we can split them and expand them until we reach passage 3 or 4, and then inactivate them and use them as a feeders to replate the human embryonic cells, or we can use them to produce conditioned medium. So basically, if we chose to froze the MEFs passage 0 cells for future usage, we have to prepare freezing medium that composed of glycerol and MEFs medium, and then we put the cells or transfer them to the liquid nitrogen. So if we chose to use the MEF as feeder cells, we need to do all further steps, and all the further steps should be carried out in a tissue culture hood and under aseptic conditions. So first thing here we need to do is to treat new falcons with 0.2% gelatin, and that will and we use passage 4 on MEF cells. Those cells cannot attach, so they will still floating. So this is slide showing the next thing that we need to do is to inactivate the MEF cells. The way that we can inactivate the MEF cells by using mitomycin C. Mitomycin C is an antibiotic which acts as a double-stranded DNA alkylating agent. It covalently cross-links the DNA, inhibiting DNA synthesis and cell proliferation, so it's mitotically inactivate the MEF cells, so we can use the MEF cells as a feeder cells layer. So here basically we need to dilute the mitomycin C and then filter it. After that, we have the passage 4 MEF cells already grow in the old media for the MEF cells, so we have to aspirate this media, and then we need to use the MEFs media that contains the mitomycin C, so we can inactivate the MEF cells. And after adding the media that contain the mitomycin C to the MEF cells passage 4, we incubate them for 2 hours at 37 Celsius. And next, after the cells inactivated, we need to wash the cells with a PBS, as is shown in here. And after we aspirate the PBS, we trypsinize the cells. And after that, we collect the cells. After we collect the cells, we add MEFs medium to inactivate the trypsin, because we don't need the MEF cells to die. Then centrifuge the cells for 5 minutes at very slow speed. And here we have the pellet cells. We resuspend the cells, the MEF cells, in more MEFs medium that contain the mitomycin C. And after that, now we can start with the first step of using the MEF cells as a feeders. Now we have the MEF cells are inactivated, so we count like we seed 56,000 cells, and from now we are not going to use the MEF medium, and we replace it with the human embryonic stem cells medium, unconditioned medium that's supplemented with the fibroblast growth factor 2. FGF2 is fibroblast growth factor 2. So this slide also showing how we are replacing the MEFs media with the HESC medium that contain the FGF2. And here, after that, we replace the media. We incubate the cells at 37 Celsius for 24 hours. And then after that, the next steps, we're going to collect all the conditioned medium. CM is the conditioned medium from the feeder flask after an incubation of 24 hours. And then we collect it in a special flask, and then add fresh human embryonic stem cells that contain the fibroblast growth factor 2, and repeat that 6 times. Every time we collect it in different flask. So we, as I said, we continue to repeat these steps for 6 days and collect all the medium and freeze them. After we finish repeating this step for 6 times, what we are going to do is combine all the conditioned medium and filter them, as we can see here. After we filter them, we store them in -80 Celsius for future use. And thank you.